And so the pandemic never stopped. And a group of people had to escape the planet Earth in search of a new planet. And hence, this group of a ragtag team of spacemen were flying across in space and they spotted an unknown planet. And because they were low on fuel, they dived in, entered the atmosphere, and crash landed on the planet. After they entered the planet, they were so amazed by the flora of the planet that they started to analyze it. We have Shireen, JVR, Sumit, Omkar, Monalisha, and Shreyas in this endeavor, and this became known became to be our LS3105 project, which we have titled Believers. Now, uh, the contents of the presentation are here, and I invite JVR Abhishek to tell us more about the introduction.
So let's start with the theory of leaf desiccation. Leaf desiccation is the process of loss of water content from leaves. During desiccation, xylem embolism, that is, obstruction and cell collapse occurs in the leaves. There is a strong reduction in cell thickness and intracellular air spaces, which results in leaf shrinkage. The leaf is not homogeneous and consists of epidermis, mesophyll, and veins. The epidermis and veins provide mechanical support to the leaf. The shrinkage is more pronounced in regions with low vein concentration. Over to Omkar.
Uh, Now we'll look at the pigmentation change and its theory. The green pigment chlorophyll present in all leaves, as we know, is very important for photosynthesis. In addition to that, there are also present the yellow to orange pigments, namely carotenoids and xanthophyll pigments. There is very less or no quantity of the blue part, namely the anthocyan. When the leaves are producing food through photosynthesis, the chlorophyll is quite stable, and due to the greater amount of it, it masks the other pigments. But during leaf desiccation and drying, chlorophyll decays rapidly in the absence of photosynthesis, and most often it reveals the underlying red, yellow, and orange pigments. Now, over to Sumit.
So JVR will now tell us about the objectives.
Our first objective was to compare leaf drying characteristics across closely related species by measuring the change in area as the leaf dries. Our second objective was to study the change in leaf coloration and leaf curvature as it dries. Over to Sumit.
Now, let us move on and see what experiments we did on this alien planet with Shreyas.
Yeah, so experiments. We will discuss this in three steps, that is, collect, analyze, and deduce. So let us see how we collected leaves. So we stepped out and explored various types of leaves available in our surrounding. As a group, we decided to sample leaves of different species, so ranging from long and thick Ashoka leaves to flat and thick Calotropis. We plucked leaves of similar size and age and headed back to our camp. Now, the next step was to take photos. In order to standardize this, we marked a rectangle of no dimension on a white sheet and placed the leaf at approximate center and took photos from a particular height. Some images were directly scanned by scanners. Further, the leaves were kept in different conditions. Some were kept in windows which were experiencing normal daylight cycle, some were in complete dark, while some were in constant light. Photos were taken on regular interval for 10 to 15 days until there was no visible change observed for 2 to 3 days. By the end of our experiment, we collected 813 images of 52 leaves from 12 different species of plants. After collection, we started to analyze. So let us first see that how we analyzed area. For analyzing area, we cropped images in the shape of the rectangle of the white paper and stored all the images in a folder. Then we created a macro in ImageJ to automate and speed up our analysis. So what this macro did was it took the image, it converted it to 8-bit image, then it auto thresholded it and converted it further into a binary image. The image was further optimized by using erode, dilate, and fill hole function. Finally, the height of the image in pixels was scaled to the height of the rectangle which was drawn on the paper, and area was measured by the analyze particle command. This way we obtained the data. Now to check our hypothesis, we plotted the area of the leaves against the time period of their drying. And like these are the graphs from two different species, and it is a bit evident that there is significant change in rate of shrinkage. Over to Sumit.
And we also had to correlate it with the phylogenetic tree. But we have to make a phylogenetic tree because we did not find any in the literature. So what we did, we used a web tool called PhyloT and International Tree of Life, or ITOL, and we used NCBI IDs of the genomes of the plants, of the some plants which we thought we had the best data of, and we used this web tools to make phylogenetic tree. And later in the deduce section, we'll see how we used the phylogenetic tree and correlated it with the area change. Now we ask Mona Lisa to talk about the color analysis.
For doing the color analysis, there were quite a few methods, but we went ahead with the simple but elaborate task of trying to find out the RGB values of our leaves with ImageJ. For that, we first made a macro similar to that made for area change analysis, which made our task easier. It gave us the main intensities of the RGB values of each leaf. These RGB values was then normalized. Since we do not have a control, normalization helped us somewhat to reduce the possibility of our data being corrupted from background noise signal. After normalization, to see how pigments change in a leaf while it dries, the RGB values were plotted against time.
Now for the color analysis, we ask Shirin to explain.
Okay, so my friends and I tried understanding how a leaf curls and why does it curl the way it does. So the very first thing we thought was, how do we analyze the curvature? A leaf is at first a 2D structure, and then as it deforms, it crumbles up into a 3D structure. And they have these curves where we get into a field of very interesting mathematics called non-Euclidean geometry, where stuff isn't on a flat plane anymore, no simple XY graphs. So we had to learn about Gaussian curvatures, but then since we haven't taken any courses like that, we wanted to try out if there was any method of calculating curl with whatever we already know. So I took different leaves like mango leaves, lemon leaves, and uh, jackfruit leaves. What I observed was, so first for the mango leaf, I thought maybe we could calculate, like imagine rolling a leaf into a cylinder along the longest beam, and suppose if you cut a slice through the cylinder, you would get a circle. The smaller the radius of the circle or diameter, the more lightly it's curved. But yeah, the leaf was uncurling in a neat cylinder, so we could not use that method. And from this observation, I understood that the vein is actually trying to stop the curl and is buckling as the rest of the leaf contracts. So I cut the leaves into half and observed that the half cut leaves, the lower part of the leaves curls up more because of the thicker vein. So now I'll show you some pictures of lemon leaves. So the lemon leaves were very difficult to handle because they were very soft and had a lot of moisture in the initial days. So I couldn't image them every day. But later, after I took a picture after 100 days, I could see in the lemon leaf there's a very beautiful symmetry of curve in left and right side, which I didn't observe for the other leaves. In the other leaves, the curls were very different in the left and right side, the analysis of which I'll tell later in this presentation. So the way I calculated the curl was, I took the leaf and I tried drawing lines from the midbeam to the edge of the leaf, and then taking as many points as I could, calculating the mean of the distances between the midrib and the edge, and taking the mean and the graphs of which I'll show you later in this presentation.
Thank you, Shirin, for the insight on the curl analysis. Now we move on to the deduction, or the part where we take the data that we have and deduce a hypothesis from it. So in the area part, we had the mammoth job of correlating this change in area with the phylogenetic tree. And we were thinking how to do that. We needed a single variable, and we found out using the slope of a linear fit of the points that we got. So what we did was we fitted the data that we have of decreasing area, fitted it to a linear fit, and found out the slope. And we hypothesized that leaves with similar slope would have similarity in the phylogenetic tree. Before that, we have to ensure that our data is statistically significant. What this means is that our null hypothesis is that there is no uh, difference in reduction of area of different species of leaves. That's our null hypothesis. And for that, we have to disprove to go further. So we used statistical significance analysis by ANOVA and the post hoc test. And we did find out that the change in area of different leaves was significantly different from each other. And so we took the pairs that were significantly different and did and the find out the slope of those statistically significant groups, and we tried to correlate it with the phylogenetic tree that we found that we made. But unfortunately, we could not find any correlation between similarity of phylogenetic tree and the change in area. This might be because of bad data, which we'll talk about later. But for now, we did not see any correlation between these two. Now, for the color analysis part, I invite Mona Lisa. But before that, I'll let me tell you about another thing that we did is is keeping the leaf in dark and constant light. And we did not see any significant difference in the change of area in the two conditions. We did significance analysis and we did not find any difference so as to speak of. So we cannot say that keeping the leaf in different environment changes the drying characteristic. Now we ask Mona Lisa to talk about the color reduction.
So, from the intensity versus time graph, we can see that chlorophyll has a reflection signal which is in the overlap of red and green spectrum. Therefore, we see a correlation in the evolution of green and red pigments. The evolution of the signal is indicative of chlorophyll degradation in a leaf over time. We can see that the green signal and by extension the level of chlorophyll remains mostly stable in the first 2 days, and then we see a spike. This spike is indicative of the initiation of mass scale degradation cascade of chlorophyll molecules. Following this spike over a period of 1 day, we get a sharp decrease in chlorophyll levels, which is indicative of complete degradation of chlorophyll. The trend is also established through the evolution of blue signal, which is indicative of the secondary pigments in a leaf. But when there is a native chlorophyll in the system, the signal from chlorophyll overpowers the secondary pigments. So upon chlorophyll degradation, there is a noticeable increase in blue signal. Now, as the chlorophyll degrades and the red pigment starts overpowering, yellowification of leaves happens. This is the point where the green and red line converge. In some cases, as we can see, they do not intersect, which signifies that there has been no yellowification that has happened yet. The graph shown next plots the green minus red values of a leaf over time, which is a parameter of how yellowification happens as a leaf dries. After comparing the intensity versus time graphs of all the leaves, we can see that there is almost a similar fate of all the three pigments in different species over time.
Thank you, Mona Lisa. But now we went on to see if there was any species-specific difference in this parameter of yellowification, that is green minus red values, the normalized values that we got. And for that, we did statistical significance analysis of yellowification of different species. And we saw that there is no statistically significant difference in yellowification, which again might be an artifact of bad data. But our data shows that yellowification does not depend on a species. Uh, there is no species specific yellowification. Now, about the curl part deduction, I invite Shirin again to talk about it.
Okay, so because we couldn't do any surface geometry, what we did was we took the leaf, placed it in a normalized box, and then took points from the first I joined the tip of the leaf and the bottom. I made a straight line and then tried measuring distances from the center to the right end, center to the left end across the whole leaf, and then calculating the mean for the left end and center and for the right end and center for two leaves across 10 days. And these are the graphs that I get. In the graphs, you can see that there is no correlation between how the leaf curls in the left end and how it does in the right end. It's very different for both the leaves, and I couldn't deduce anything from these graphs.
Thank you, Shirin. And now for something extra that we did in our free time, and Omkar will tell us about it.
So, in addition to all these varied experiments and conclusions that we drew from the analysis of the leaf color, area, and curl, we tried, let's get back to our own earth and try to look at some real data. That is, we tried to look at some forest deforestation images available freely on the internet. For this purpose, we took some images of a province named Nova de Chaves in Bolivia, and as you can see, the deforestation is quite evident from this slide. The a series of images of this particular region were taken from 1984 to 2018, and which shows a significant deforestation. And in order to know how much deforestation happened, we tried to quantify it in order to by detecting the deforested area as well as the forested area. In order to do that, we created took screenshots of the year satellite images of the particular forest area in Bolivia, and then a macro was created which thresholded each image and adjusted the brightness and contrast so that the forested areas, namely the green areas, could be detected, while the non forested yellow areas were relegated to the background. We then performed object analysis on the thresholded images in order to find out the area of the forest by analyzing each particle. Following which, we plotted the simple area versus time year graph as it is shown in this slide. In this graph, we can see that the forest cover has been constantly decreasing over uh, the years, and that the rate of forest deforestation that is indicated by the slope has actually accelerated from the year 2000 onward, which has coincided with a rapid urbanization and increased incidence of forest fires in the region of Bolivia that we are talking about, and which is a very worrisome detail. So this was something that we tried to do in a free time. Let us now get back to our main things.
So thank you, Omkar, for this interesting tidbit. And now we move on to conclusion, for which I ask again Abhishek Javia to tell us about the final conclusion we derived from all these experiments and analysis.
We found that the drying curves of the leaves were species specific and was not related for closely related species in our sample space. We did not find any significant relation in the yellowification and species. We did not get any good conclusion on leaf curling and the species. We found that the drying characteristics of leaf kept in day-night cycle and dark cycle was not significantly different. We found that consistent deforestation in Bolivia occurred across 35 years. Next, let's look at some of the mistakes we did during our experiment. For area analysis part of our experiment, we had initially used the leaves that were pressed under books. After discussing with Visha ma'am, we realized that the pressing the leaves non-uniformly using the books affected the drying rates, and hence did another batch of imaging without pressing the leaves with books. Due to the scarcity of time, the sample size of this second batch was small, and we had to include the first batch as well in statistical analysis. Now, for color analysis, the sample size was small, and we couldn't take many images again due to the scarcity of time. Then, for the curvature analysis, As the leaves were curling, there were cases where the shape of the leaf was such that the leaves could either be placed over the left side of the midvein or the right. This means that the orientation of the leaf across time could not be kept fixed. This could be avoided by taking images from multiple angles and including them in the analysis. Over to Sumit.
Thank you, Abhishek, for discussing all these mistakes and the errors that could have been there in our experiments. But even if all this mistake and failures that was very consistent, we did have a lot of fun and we learned a lot of things. And for that, we'd like to thank Dr. Bidisha Sena, who gave us this opportunity to not only learn a lot about imaging, but also a bit about how a hypothesis is formed and some basic statistical analysis. We'd also like to thank Gaurav from Department of Physics in our own batch and our seniors Anshuman and Amrutmay, who helped us with our analysis. And this was our group, and we met regularly to discuss and do the experiments. And for now, we blast off back to our home planet to see if the pandemic has been over or not. Until then, we'll move around in space in looking for new planets to explore. Thank you.